Now the fast.
Um, you need this. I think I need this. First of all, I'm here because Jamie asked me to do, and I'm doing whatever Jamie asks me to do. Second, thank you guys for this conference. It's been really amazing, and it's just so exciting to be part of it. Thank you very much. I'm representing here AFAR and one of the projects that we adopted that we called FAST. And FAST is, if I rephrase what Andrew said about ARPA, it's a solution. And you just heard the problem. It's one thing that we're making such a big advance in discovering biomarkers for aging, biomarkers for biological aging. But really, this is not what we need to do. We need to find the biomarkers of aging that are changing with interventions. Why do we need a solution for that? First of all, because our clients are people who now are measuring their cholesterol or glucose or hemoglobin A1C and are making decisions. We need the people to join us, and they have to know if they're making progress and if they're making progress in a relatively short period of time. Second, XPRIZE or biotechs need to know in a phase 2 trial if we're doing something significant in aging so that we don't go and lose $100 million in a phase 3 trial. And third, we want pharmaceuticals to understand that there's a way to discover what they're doing quite rapidly and make their path through FDA approval much better, okay? This is what we need solution for. And I'll tell you what I think and why I'm thinking, and I'll be interested to know what you're thinking. But if we're talking about gerotherapeutics, the first thing that we want to know is that they're affecting the hallmarks of aging. Oops, sorry, I didn't mean that. I thought I might be using an old set of slides. But anyhow, the hallmarks of aging. And the interesting thing of the hallmarks of aging that are interrelated to each other, so that if you target one of them, you affect the other. This is a really good thing for us. And it happens with gerotherapeutics. You know, statins have a huge effect on health, but they're not changing the hallmarks of aging. And they don't cause our animals to live longer, okay? It's only cardiovascular specific. So we want to see if they affect, from a preclinical study, we want to see if they affect the hallmarks of aging. Also, I should say, you know, if they're interconnected, I could target metabolism, one hallmark, and affect immune decline or inflammation, another hallmark, okay? So keep that in mind. The second thing preclinical, we want to see that the drug that we're using has affected health span and lifespan in animals, or as you'll see, you know, if it's possible in humans, but in animals, okay? That's how we are really supporting our field and saying that we're doing something substantial that is worth clinical intervention. From a clinical perspective, we want a 3 years or a 5-year trial that will show that the drug that was designed for something is actually preventing several other diseases, other multi-morbidities. In a clinical trial, placebo control, we also want to show that a certain gerotherapeutic, not effective only the disease and the disease-specific mortality, but affected overall mortality, suggesting that it's really somewhere in aging. I would say if you could show the 4 of those, you actually have a gerotherapeutic. Oh, that's where my thing have gone. It's the interaction between them. Anyhow, so are there such drugs? Who thinks there are such drugs? Raise your hands. Okay, I'm glad it's the minority. It's not going to be boring for the others. So we, and when I say we, is huge amount of investigators that have published 2 papers till now, and I'm just showing you part of the second paper, where we gave basically 6 points for the preclinical part and 6 points for the clinical parts. And we took drugs that are A, FDA approved, B, have shown at least in one model to extend lifespan in animals. That's how you get in. There could be many other drugs like that, but those are the drugs that FDA approved and have changed longevity. And you see that there are 4 drugs that got over 10 according to the scale that we've done. And I should say that GLP-1 is changing places by now. Bisphosphonates is going down, but all of them are going to be 11 points out of 12 points, okay? You notice that rapamycin is not one of them because rapamycin doesn't have good clinical studies to make us sure that we can use them now. Another point is that at least some of those drugs have shown in clinical trials that although they're giving to one indication, they also prevented mortality, hospitalization, and long COVID, okay? Basically connecting, those are not only metabolism or bone or something like that, they also affect the immune system or resiliency to deal with severe disease. So what are the evidence? So I'll give you, first of all, a side to side. This is the ITP with SGLT2 inhibitor with the CANA. The top parts are, you know, it's 3 centers. So let's look at the top, which is the average for the 3 centers. And it's female where you see no effect, and male where you have a significant increase in lifespan in all the males. And let's look now at the studies in humans. The studies in humans that included more than 4,000, this study only, there are many studies, by the way. This study included 4,000 people, more than 4,000 people, and the cardiovascular outcomes in 32 months, right? It's 32 months, 32 months, decreased by more than 30%, the renal by more than 40%, and overall mortality by 30%. Okay, by the way, longevity in clinical trial is really decreasing mortality, right? That's all we can do. In other words, I'm showing you a drug that has extended healthspan and lifespan in animals, and that in human trial was designated for diabetes. By the way, most of the people who take this drug are not diabetics. Those are people with renal or heart indication. They're the ones who were getting that because this drug affects significantly and in a very short time other diseases. I'll go really fast for healthspan outcome for each one of them. SGLT2 in another study with 1,800 people decreased cardiovascular and renal outcome by 28%. Metformin in heart failure decreases outcome by 30%. Biphosphonates for CVD decreased by 33%. And GLP-1, the SEMA-1, 22% for renal outcome. I don't know if you knew it, but GLP-1 does many other things beside obesity. And the same thing is for mortality. Decreased mortality by 32%, SGLT2, metformin, 24% in diabetes with atherosclerosis, biphosphonate, 50% in people in ICU, and GLP-1 decreased mortality by 50% in several studies. I can go on, and that's a paper that we're preparing. Some of you guys are authors in those papers, but there are 4 drugs that have showed incredible data to show, yes, we already have gerotherapeutics, and it has been shown to be effective in clinical study. If you wanted to actually do a clinical study, you would design this study probably. The advantage of using those studies is that if we want to get them into the public, we want MDs to do it first. Okay, supplement, everyone can tell somebody else to take supplements. Those are drugs that are prescription, and we don't want to kill anybody on the way to success. So we want to have MD involved, and MDs have the safety, the efficacy, and the other data that was accumulated during the year. But more important, it's legal to repurpose an FDA-approved drug because FDA-approved drug is safe and monitored and followed It's legal for any physician to do that. That's not absolutely a crime. As an example, metformin was used for obesity, not the best drug anymore. It's the first drug of choice for polycystic ovary syndrome to prevent prediabetes. Metformin is prescribed in Europe mainly to people who have COVID to prevent hospitalization and mortality. None of that is FDA approved, but people are using metformin, and people are using metformin for aging also. So why am I telling you all that? First of all, that you should know, we have those drugs. Anybody can have the drugs because you have clinical data to suggest we're there already. But from the biomarker perspective, we want to use those studies that are already ended and do the omics in as many as we can. And for that, by the way, Jamie and I were leading this project. She was demoted to the X Prize. And now it's Dan Belsky and Madi and I that are driving this. And you see that other people there, some are here in the audience, are in the SAB of that. And basically, what we've been doing, we're conducting those studies. We're taking 3 samples from before, one sample from before, and actually 3 afters we have for most of them. And we're starting to accumulate those samples so we can do the right omics in the last time, in the right time. Now, this is what's going to happen. Each of those drugs is going to have an effect of the drug itself. Okay, the drug is going to be something unique for the drug. But also, those drugs are going to be something that are specific for aging. And it's this center, it's the overlap of the biomarkers of those drugs that are common to everyone, is the biomarkers that we want to, as fast as possible, to get on, to make available for everyone in pharmaceuticals, so we can get ahead and really accelerate the field that so much depends on us. I want to make just one point. That's my last slide. We are writing, the paper we're writing now is, okay, we have 4 drugs. If you want gerotherapeutic, how do we know which is good for you? And look, we know already for every drug that we can save 100 and kill 3, right? This is true for every drug. The question is, how do we choose the right 100, and how do we avoid the other 3? Biomarkers will certainly help us do that. But what we can do now is go to those studies, because those studies have sub-analysis. Those studies says, what happened to people over 65 and less than 65? What happened to men versus women? What happened to other things? And this is not complete. You can take picture, but this is not complete. And many things are not studied, but you see that there are a lot of effects, and some effects that have been shown specific for some of them. I mean, all of them are preventing cardiovascular disease. Some of them are good for renal. Some of them are good for inflammation. One of them is good for Parkinson's. Some of them are for NASH. So we want to do a secondary prevention. In other words, we don't want young people to get those drugs. We want old people who already showed sign of aging, maybe had a disease, something like the TAME people, to say, you know what, we can do better, and those drugs are available to you. And I think that's the end. I'll stop here. Thank you.
We have about 1 minute, so there's time for one fast question. It was a bad pun. Nobody caught it.
Any reason you left out ACE inhibitors? Beta blockers, perhaps? Some of the other cardiovascular drugs, because you're really concentrating on cardiovascular drugs.
2 things. First of all, they were on the list, okay? ACE inhibitors were in the list. They didn't score high. But more important for this project, those ACE trials have been, were 20 years ago, and we don't have an access to the plasma. The once old study that we have access is was called the diabetes prevention trial. The diabetes prevention trial, we took people who were not diabetic, but at risk for diabetes, and treated them with lifestyle, with metformin, and with TZDs. TZDs was stopped after a while. The samples are at the NIH, and they're transferring us parts of the sample for 2,800 people. So we'll have a lifestyle and other from old studies, but we're not aware. And by the way, if you know of any study, if you know of any clinical studies, big or small, that could contribute here, we would love to know that, because I think we're going to expand. We have some funding now. I think we're going to expand it dramatically in the next few months or years.
All right, I think that's all the time we have. You can catch Nir after.